Plasmids. Plasmids are extrachromosomal supercoiled double-stranded DNA which is used in transferring of various genes. For example, plasmids are self-replicating and plasmids are extrachromosomal elements which are present in a cell. That is, rather than a genome or a chromosomal genome, we will have a part known as plasmid. This plasmid also will have various elements and genes which is responsible for various benefits to the cell, but it is not necessary that it is going to keep the cells for the survival. So it is not an essential part of a cell. So moving on to the plasmids. So if you take a bacterial cell, you will have a bacterial genome that is double-stranded. You also have a circular supercoiled double-stranded DNA. So this is known as the plasmid. So this is the bacterial DNA and this is the supercoiled double-stranded DNA, that is plasmid. The main few properties which is gonna observe in the plasmids are they are extrachromosomal material. The second property of the plasmids, for example, if you take the plasmids are extrachromosomal material, and the main property of the plasmids are plasmid. Plasmids are extrachromosomal, small, circular, supercoiled, double-stranded DNA which has been present in a bacterial cell other than that of the chromosomal DNA. So these plasmids are not gonna affect the chromosomal DNA, and it also contains some benefit features which has been present in the form of genes. This is gonna help the bacteria, but it doesn't mean the survival of the bacteria is decided by the plasmid. There are various types of plasmid depending upon the function. The main three properties of your plasmids are they are extrachromosomal material, that is, rather than of a chromosomal DNA, then extra chromosomal material which is present in the DNA, present in the bacterial cell is termed as plasmid. Besides, they are self-replicating, that is, they are not dependent upon the template strand which is already available, that is a chromosomal DNA. It is self-replicating. The third one is it is used for conjugations, and it is contains genes which are non-essential. It is not used for conjugation. It is used for conjugation, and it is not contains genes which are not essential for the process, that is for the survival of the bacteria. So this is the structure of the plasmid. A bacterial DNA and a supercoiled double-stranded DNA, that is a plasmid, is present. So plasmid is a extrachromosomal double-stranded supercoiled DNA. It is an extrachromosomal material, it is stated because rather than of a chromosomal DNA which is present in a bacterial cell, there is an extra chromosomal DNA which is present known as plasmid. They also contains genes which are responsible and which are helpful for the benefit of the bacteria. But it is not gonna nowhere gonna disturb the chromosomal DNA, and it is nowhere gonna harm the bacteria which has been harm the bacteria where the plasmid is present. The other important feature of your plasmid is it is a self-replicating. That means it is not gonna depend upon the chromosomal DNA for the replication process. So the number of copies of the plasmid also will be decided depending upon the replication process. So there are other two terms which we commonly come across when we are learning about the plasmid is copy number and compatibility. Copy number determines the number of plasmids which is present within a single cell. So number of copies will vary. For example, if you take a very large plasmid, then there will be number of copies of the plasmid is very less because the bacterial cell cannot accommodate large number of plasmids which is in larger size. But if you take a plasmid which is in very smaller size, the bacterial cell can occupy larger number of copies, that is high copy number if the smaller size of the plasmid, and larger copy number and smaller copy number if the larger cell size is very large. So this is vice versa. Compatibility comes like if a plasmid of two different bacteria, for example, a bacterial plasmid from a Pseudomonas and bacteria from an Agrobacterium is to combined in a new bacterial cell, the compatibility of the two plasmid to stay together in a single bacterial cell is termed as compatibility. So these are the two distinct features of a plasmid. Now we are going to look onto the different types of plasmids. For example, there are two main plasmids we come across, that is conjugative plasmid and non-conjugative plasmid. We call it as plasmid which is useful for conjugation and the plasmid which does not contain a pilus or a sex factor, fertility factor, which is not gonna help you for the transferring the genes. So they are known as non-conjugative plasmid. So based upon the conjugation, it is conjugative and non-conjugative. The second type of plasmid is integrative and non-integrative. For example, if a plasmid is carried from a source and it enters into another cell and where it is gonna replicate and produce larger number of copies. But there are other other source. These are known as non-integrative plasmid. The next category is the integrative plasmid. Integrative, in the term itself there, integration process. So what happens is, like when a plasmid, it is gonna carried out, a single strand of the plasmid is gonna worn out and produce a double stranded in the other source cell. So from the host cell, it is gonna go to the guest cell, can consider. So once it is gonna go there, so donor to the host cell, once if it is going there, it is gonna integrate with the chromosomal DNA of that bacteria. That means from the donor, it has been gonna carry, that is, it is plasmid is gonna move that into the host cell to the conjugative tube. Once it is gonna move there, it is gonna integrate with the chromosomal DNA which is already present in that bacterial. So it is gonna disturb the chromosomal DNA. That means it is gonna integrate and produce a double strand. So some features of that plasmid is gonna get incorporated into the chromosomal DNA of the host cell. So this is one known as integrative plasmid. And other types of plasmids are based upon their work or the function it has been done. For example, if it is used for carrying, like conjugative plasmid is also known as fertility plasmid. The main thing there is the fertility plasmid will have a transfer genes, that is TRA genes. So this transfer genes is responsible for carrying the genetic material from that host cell or your donor cell to the host cell through the F factor, that is known as with the transfer genes. So this is known as fertility plasmid. The second type of plasmid is known as Pseudomonas plasmid. These plasmids are containing the material with the, like a genes coding for antibacterial infection. So if you introduce this plasmid to your source, it is gonna show some make some antibacterial protein through the antibacterial genes, and it is gonna act against the antibacterial. Next comes the resistant plasmid. Resistant plasmid, in the name itself, they have a resistant genes within the plasmid, so it is gonna resist against a certain bacteria. So if you want to act against certain bacteria, we can use those bacteria with this plasmid, so resistant genes. Next, the fourth type comes the Col plasmid. Col plasmid, the name itself there, Col will produce a protein known as colicin. It is also a type known as bacteriocin. This will act as agents for killing other types of microorganisms. Next comes degrading plasmid. Degrading plasmid is like, for example, if you take oil spills somewhere, so you're gonna add Pseudomonas butida, like the commonly available superbug. So Pseudomonas butida will have a plasmid that will have a genes coding for degrading the oil spills which is there in the surface. So the plasmid is coding for the genes that is known as a degrading genes which is gonna degrade the oil spills. So it is not the DNA which is gonna take care of it. It is a plasmid which is gonna take care of the oil spills. So that's why it is known as plasmid, degrading plasmid. The next category comes the integrative, non-integrative fertility type. So depending upon all these types on the function, plasmids are defined. For example, the last category will be the virulent plasmid. The plasmid itself is termed as virulent because it contains a virulent genes. So the virulent genes is gonna make your pathogen virulent. So we call the plasmid with the virulent genes are known as virulent pathogen. So we're gonna look that into the detail in this section. So plasmids, bacterial DNA, supercoiled double-stranded DNA. So this is termed as a plasmid. If you take a typical structure of a plasmid, for example, I am drawing a structure of a plasmid known as plasmid BBR, PPR. So this is nothing but, so this plasmid is named after the person who has identified this plasmid. So it is known as plasmid Bolivar and Rodriguez. So this plasmid is gonna have, like I already mentioned, they're gonna have some restriction sites. You can take other color. This is gonna have a restriction sites. So at least a plasmid will have at least 2 to 3 restriction sites. For example, the major restriction enzymes EcoR1. You can write BAMH1. Then you can write SAL1, EcoR1, BAMH1, SAL1, etc. It also got a site we can claim as, this is antibiotic, that is tetracycline resistance genes, that is antibiotic resistant gene. Example, tetracycline resistant genes. So usually saying in the name of like ampicillin resistant APR, TETR. So these are the examples for antibiotic resistant genes. Then you will have a main site known as origin of replication, where your bacteria, this bacterial plasmid is going to replicate. This is a site where it is going to replicate. It is usually termed as ORIV, origin of replication. Origin of replication. Other than that, you're going to have transposon site, TRN site, which is going to transposon site. Transposon site. Then you also have insertion site, IS1, IS2. This all denotes the insertion site. So this is a typical structure of a PBR322. So they gonna have some example, like they gonna have some antibiotic resistance gene site, for example ampicillin resistant, tetracycline resistant, followed by a origin of replication, where your replication is gonna get initiated. Then you can also have a transfer gene site, that is transposon site or transfer gene, which has been present in your plasmid, which is gonna help you in the attachment and transferring. And next comes the insertion site, which are the sites which are capable for insertion to then another plasmid, that is insertion 1 site, insertion 2 site, marked in the form of IS. So it's a typical structure of a plasmid. So the main properties, the first properties of a DNA is it is an extrachromosomal material. It was introduced by Joshua Lederberg in 1952. The second one is it contains genes like TRA, IS genes, all which are non-essential for the survival of bacteria. Third one is self-replicating. Next one is the fine thing, the size will vary from 1 kb to 1000 kilobase pairs. It is also has other names. The other names are like sex factors, conjugates, extrachromosomal replicons, also called as transfer factor because it contains TRA genes. So these are the typical structure of a PBR. Then you comes the basic features, that is extrachromosomal self-replicating, size from 1 kilobase pairs to 1000 kilobase pairs. Other names for the plasmids will come under this property. Then I've already mentioned what is copy number, the number of copies of plasmid which are present within a single cell. Next comes the compatibility, the compatibility of the two distinct plasmid to survive at a single bacteria. So copy number, high copy number, low copy number. For example, larger the size of the plasmid, then only lesser copy number. Smaller the size of the plasmid, higher the copy number. Compatibility, you can take two distinct plasmid surviving in a single bacteria without harming each other or acting against each other, that you can also term as compatible to each other. This is termed as compatibility. So these are the properties we are going to see in plasmids. Next we are going to discuss about the different types of plasmids. So this is the donor cell, this is the host. You will have already a chromosomal DNA which you can see. This is the extra chromosomal material present. If extra chromosomal material is present within the cell, it is known as the F plasmid. So what happens, it is capable of forming a pile that is known as conjugation pellets. These conjugation pellets will draw the cells together. So the cells will come in close with each other. So what happens, first coming to close each other, a wall will be open. Second one, a single stranded structure of the plasmid goes into the next plasmid cell. So where it could go and replicate to form another plasmid. So this plasmid which has lost a single stranded also will restore the plasmid, that is a second strand of the plasmid, by replication process. So both the cells replication of the plasmid takes place. This is how plasmid is integrated into another plasmid, or it has been transferred into another plasmid. So this is the first one based on the conjugation. Now based upon the copy number. Based on the copy number, first one is the non-integrative plasmid. Non-integrative plasmid. So under non-integrative plasmid, for example, conjugated non-integrative, here have non-integrative and integrative. So under non-integrative, in this type of replication, what happens is like plasmid gets entered into the source, that is into the host cell. From the donor, plasmid gets moved down into the host cell, where the plasmid gets replicated and produces the number of copies of plasmid. But in integrative plasmid, what happens is like plasmid enters into the host, integrate with the chromosome which is present within the host cell. That's why it is known as integrative plasmid. So non-integrative plasmids are plasmid in which in this type of replication, the plasmid DNA once introduced into the cell, for example, it is getting introduced into the cell as per the copy number and the multiplication. So it's already will decide how much copies number, so accordingly it will undergo multiplication. But in integrative plasmid will integrate into the bacterial chromosome. Such plasmids are also known as mobilsome. We call it as episome or mobilisome or episome. They replicate along with the bacterial chromosome. So this is the main feature of a integrative plasmid. So normally plasmids are self-replicating. So if, for example, if you take integrative plasmid alone, they're going to integrate with the plasmid, integrate with the chromosomal DNA which is present in an host bacteria. So we're going to see the integrative and non-integrative plasmids. For example, this is a non-integrative plasmid example. This is the episome, that is the integrative plasmid. Already you will have a bacterial DNA here. It's a bacterial chromosome. So there is, the process is done. Here the plasmids, these are the plasmids. These plasmids enter into another bacteria through conjugation, that is F-fertility, which has already been discussed. Once it enters, it maintains the same copy number, and this is known as the cell division. Here the cell division process takes place. Just the cells get divided and equally the number of copies of the plasmids also remains the same. But in episome, what happens is like the plasmid which is present in the bacterial cell, that is the donor cell, it is going to get integrated into the bacterial chromosome. So then it is going to produce a structure which is similar to that of the bacterial chromosome as well as the plasmid. So you will have integrated plasmid that will have bacterial chromosome, chromosomes plus the plasmid factor, and they replicate together. And next category, that is now we have seen so far two categories of the plasmid. The first category of the plasmid is conjugative and non-conjugative, followed by integrative and non-integrative. Now we are going to look into the types of plasmid based upon their function. Third category, classification based on functions. Based on function of plasmids. So the first category is the fertility plasmid. As the name indicates, it is used for the fertilization. That is, F plus factor will be present. So that contains it contains the transfer genes, the main feature, which is gonna transfer your material to the next gene required for conjugation. Else known as F plasmid. Fertility factor, that is transfer genes. It is known as the F plasmid. Example, F plasmids in E. coli. F plasmid, if you take the example, E. coli. The next second category comes the resistance plasmid. So as the name indicates, it is gonna carry a resistor genes within the plasmid. So this resistant genes is gonna carry genes of resistance to it is gonna carry genes which is resistant to avoid one or more antibacterial agents. So if you want to kill a microorganisms, you're gonna add some antibacterial agents. The plasmid with the resistant genes is gonna inhibit the activity of that antibacterial agents. The third category is the Pseudomonas plasmid. In this type of plasmid, it is used for the treatment of bacterial infections. So for example, if a bacteria is causing an infection, the Pseudomonas plasmid can be utilized for the acting against that bacteria which has caused the infection. So you can write help in the treatment of Fourth one is the Col plasmid. As already discussed, Col plasmid is gonna produce a protein known as colicin or bacteriocin. This is gonna act against the other bacteria. This bacteriocin will kill the other bacteria. So this proteins can be produced from this plasmids and can be utilized. So this is a byproduct of a bacteria which contains the Col plasmid. Bacteriocin or colicin. So this bacteriocin and colicin is gonna act against another bacterial agent. So this is also act as an antibacterial agent which is produced by a plasmid which codes for Col genes. Next comes the fifth category, degradative plasmid. So it is gonna code for a degrading gene. And that is gonna act on that example the degrading material. For example, you can take it as a metal. For example, or a chemical. For example, if a plasmid is coding for a gene which is going to act against a specific chemical, for example, you can take toluene, then the gene name will be TOL gene. So TOL gene will be responsible for degrading the TOL gene which is present in that plasmid, which is present in that bacteria, will act against the toluene chemical which is present in the soil. So for example, TOL gene of Pseudomonas. So this is the best example for degrading enzymes. Degrading bacteria acting against Pseudomonas. Next comes the virulent plasmid. As the name indicates, it gonna contain a virulent gene. So virulent gene in the plasmid which is gonna make that bacteria itself a virulent. So you call it as a virulent pathogen. For example, you can take TI plasmid. TI plasmid of agrobacterium tumefaciens is gonna act against the or cause ground gall disease in dicot plants. So it is one of a plasmid which is a virulent plasmid which is gonna make that bacteria itself a virulent, and it is gonna acting against the plants, that is the dicot plants. So TI plasmid of agrobacterium tumefaciens, which is gonna cause ground gall disease in dicot plants. Now, other two types, which is a very common type of a plasmid, is relaxed plasmids and stringent plasmids. Relaxed plasmids are those plasmids in which the number of plasmids, that is the copies, are maintained in a still. That means it cannot be changed. Stringent plasmids are those plasmids in which the copies remains very less. So this is a rare group. So we can call it as relaxed plasmid. So normally the plasmids are maintained at multiple copies per cell. But if we come across the stringent plasmid, limited copies per cell. So limited copies, multiple copies. So this is another category of plasmids. And finally coming on to the category how plasmids can be confirmed. So normally we go for a gel electrophoresis process, and its size marker is from 1 kilobase space to 1000 kilobase space. So based upon the form, how they are formed, they gonna look in the gel apparatus, that is under UV conformation. You're gonna look at the size, and based upon that you're gonna confirm the plasmid. For example, if it is a linear plasmid, then the size of the structure will be very thin, and if it is a single stranded plasmid, it is gonna look much more thinner than that of the linear stranded plasmid. If it is a double stranded plasmid, it gonna look like a thick band. So the size also and the molecular weight of the plasmid also varies. If it is a supercoiled structure, then there will be, you're gonna look at the inner curve in the band which you're gonna get under plasmid. So this is how conformation has been carried out. Conformation is usually carried out by gel electrophoresis. So under gel electrophoresis, if you look, you will have a conformation, that is first one is, you can call it as nicked open circular DNA. That is DNA has only one strand. And you will have a linear. The second category is linear DNA that looks a little more thinner. Third one is supercoiled. We also say covalently closed circular DNA. Then last you have supercoiled denatured DNA. See, these are the some types of bands which has been seen after a plasmid is visualized under a UV electrophoresis. So under UV gel documentation, if you look at, these are the few types of plasmids, how they gonna differ. For example, nicked linear supercoiled, that is covalently closed circular DNA, finally comes supercoiled denatured DNA. So they gonna appear. For example, if you take, this is the gel electrophoresis under UV. The thin one, we can call them as nicked. It is a single stranded DNA. Then if you take a little more thick, this is called the linear. Under the thick band, we can call it as covalently bended. Next you can have supercoiled. Supercoiled will look like this. There will be a bend at here. A covalently ended means inside there will be a covalent bond which is gonna seen out in the band. But if you look at the supercoiled band, you can see the coiled structure of a plasmid. Circular and single stranded DNA will appear very minute, very thin structures will have it as a single stranded DNA. There are some type variations in how a plasmid is viewed under UV gel documentation. So looking on to the applications of plasmids, if the plasmid is kind of used as a carrier molecule, so for example, if you want to transfer your genes, you can carry to the plasmid to another bacteria or another source where this can be used. So it can be used for gene therapy. Another one is can be used for studying various disease, like a disease models also this can be used. Agrobacterial tumefaciens, it is a wide variety of plasmid, TI plasmid, which is studied and utilized in agricultural engineering. So these are some applications and functions of plasmids, like it is used as a disease models for various disease studying. And another one is TI plasmid in agricultural engineering, and it can be also used for coding the function of the gene and then finding what gene, what protein is being produced through the plasmid. So it is useful for carrying from one host to another host. So this can be used for gene therapy application. Thank you.